Good morning, students. Hope you'll be fine at your respective places. Today, we are going to discuss the structure of mRNA, that is the messenger RNA. We have already discussed that we have got on the basis of their functions, we have got three types of the this RNA. First one is the mRNA, then we have got the rRNA, then we have got the tRNA. And today, we are going to discuss this mRNA. Now, what do we mean by this mRNA? Yeah, हम ये कहेंगे why it is called as mRNA. This mRNA, simply, it consists of genetic information. This mRNA, it consists of genetic information. That genetic information is copied it on by this DNA, and mRNA uses this information and uses this information to code a protein. So what we will see, this mRNA, why it is called as a mRNA? Because it consists of genetic information, and that genetic information has been copied on by DNA, and this information has been copied to it by DNA. वो इंफॉर्मेशन यहाँ को DNA से आ गई है, एंड mRNA जो होता है वो इस इंफॉर्मेशन को यूज कर लेता है टू कोड ए सर्टेन प्रोटीन, एंड that is why this mRNA is present within the nucleus. It is present, it is found inside the nucleus. Now, the formation of mRNA from one strand of DNA. We know that DNA is a double strand. Now, the formation of mRNA from one strand of this DNA is called, this process is called transcription. So we will see this transcription, that the formation of mRNA from one strand of DNA, because we know that DNA is a double stranded, from one strand of DNA, that is called as transcription. We know that the DNA is double strand. जो DNA हमारे पास है, that is double stranded. लेकिन when replication takes place, that is the breaking of DNA takes place, breaking up of this strands of DNA takes place, that is called as a replication. That means when replication takes place, what we will see, both these strands, they separate from each other. वो एक दूसरे से अलहदा हो जाते हैं, and that replication, when this separates from each other, that give rise to the formation of this mRNA. I will repeat this. We know there are two strands of DNA, and when replication takes place, when breaking of these strands takes place, both the strands, they, what we call, say that both these strands, they takes apart, वो एक दूसरे से अलग हो जाते हैं. But when we see, when that gives rise, and only one of these strands, it gives rise to the formation of this RNA, which is called as the mRNA. So this mRNA, it is formed from only one strand of the DNA, and it is found within the nucleus of the cell. And from the nucleus, it is shifted to this cytoplasm. From nucleus, this mRNA, it is shifted to the cytoplasm, and in the cytoplasm it combines with the ribosomes. In the cytoplasm it combines with this ribosomes to form the polypeptide chain. It forms the polypeptide chain. So this mRNA, simply, it acts as a template. It acts as a guide. This mRNA, it acts as a template, or we can say in layman's language, it acts as a guide, as it has genetic information. This mRNA, it has got genetic information, and this genetic information, this template or this guide, it acts as, thus it acts as a template, it acts as a guide for the synthesis of protein. For the synthesis of protein, this mRNA, it acts as a template, or in the layman's language, it acts as a guide, and it contains the genetic information, and that template is used for the synthesis of the protein. So if we write the equation, what we write? That we have got the DNA, and this DNA, what we will see, it gives rise to the mRNA, and this process takes place by means of transcription, and this mRNA, it gives rise to the protein synthesis in the ribosomes, and that is called as, that process is called as, we can study it as, we call it as the translation. Now we know that this mRNA, it contains the genetic information. This mRNA, it has got the genetic information to code as certain, to code a particular, what we call as amino acid. To code a particular amino acid. And on mRNA, what we will see, there is the genetic information, and that genetic information जो उसमें होती है to code a certain amino acid, that genetic information is in the form of codon. That is in the form of a codon. Now the, what we call as the sequence of these codons, the sequence of these codons, they determine the sequence of the amino acids. So we can write it like this: sequence of codons, it gives rise to the sequence of amino acids. So we say the sequence of codons, they determine the sequence of amino acids in a protein chain. In a protein chain. Now, if we see, what we call as the two strands of DNA. If these are the two strands, rather, if we are the two strands of these DNA, now we will see these are the two strands of the DNA. This DNA, this strand rather, not DNA, this strand of DNA, it contains what we call as this strand. It contains the genetic information in the form of codons, and this is called as template. This is called as template strand, or we can say it is called as the guide strand. And this guide strand or this template strand, it takes part in the formation of mRNA. Now this part, it is called as coding strand. This is called as coding strand. Now what we will see, mRNA जो होता है, it is formed in the direction of 5 primer in the direction of 5 primer 3 primer direction, in this direction from 5 primer to 3 primer direction, ये याद रख लेना. Now this part which contains this template part, and this part which contains the genetic information for the synthesis of a polypeptide chain, that is called as cistron. This part, this is the cistron. So what is called as this cistron? This is the template strand. This whole is template. Now this part, यहाँ से यहाँ तक, which contains the genetic information for the synthesis of a polypeptide chain, this is called as the cistron, and this is called as the cistron. And this cistron, it is what we call as the similar like that of the gene. Cistron is someone like that of the gene. It is structural, it is the functional unit of this DNA. Now, अगर हम बात कर लेंगे this mRNA, it is, इसके कुछ characteristics हैं mRNA के. First one, it is short-lived. Okay. It forms one or two these amino acid chain, then it dies. So it is a short-lived. And दूसरी बात, it has got the molecular weight that is very much high, very high molecular weight, that is about 5 lakh to 10 lakh. Lakh to 10 lakh. So it is molecular weight is very very high. थर्ड, अगर हम इसकी कैरेक्टरिस्टिक फीचर देखेंगे, इट इज फाउंड इन लीस्ट क्वांटिटी, नियर अबाउट 1 to 5% ऑफ इट इज ऑफ द टोटल आरएनए जो है, ऑफ द टोटल आरएनए जो है, दैट इज mRNA. So we can say it is short-lived. Its molecular weight is 5 lakh to 10 lakh. It is found in the total mRNA. Total RNA में अगर मैं देखेंगे, mRNA is least quantity, about 1 to 5%. Now this mRNA is of two types. अगर हम मैसेंजर RNA की बात कर लेंगे, we have got two types. एक को हम बोलते हैं मोनोसिस्ट्रॉनिक mRNA, दूसरे को हम बोलते हैं पॉलीसिस्ट्रॉनिक mRNA. This monocistronic mRNA, it is mainly found in what we call as eukaryotes, and this is mainly found in prokaryotes. Monocistronic mRNA, it is mainly found in eukaryotes, while as this polycistronic mRNA, it is found in prokaryotes. As the name indicates, that means this what we call as monocistronic mRNA, it has got the genetic information, जो हम इसके बात करेंगे, genetic information जो इसमें पास पाई जाती है, genetic information जो इसमें पाई जाती है, it is not more than 1 polypeptide chain. Peptide chain. अगर हम यहां पर देखेंगे, जो जेनेटिक इंफॉर्मेशन पाई जाती है, जो हम कहेंगे इट इज व्हाट वी कॉल एज दिस ओनली 1 पॉलीपेप्टाइड चेन, ओनली इट इज फॉर मोर देन 1 polypeptide chain. So monocystronic में हम बात कर लेंगे. It has got the genetic information and that genetic information codes for only one polypeptide chain, while as in case of the polycystronic, it is that it contains the genetic information for more than one polypeptide, what we call as the polypeptide chain. Now, if we study the structure of the mRNA, it is thread-like. It is thread-like, it is long, and it is a single-stranded. It is a single-stranded. So if we study the structure of it, is a thread-like, it is long, it is a single-stranded, and it is mainly found in the direction of 5' to 3', like this. 5' to 3' end, and it is found in the direction of 5' to 3'. Now, if we draw the structure of this mRNA, see here, if we draw the structure of this mRNA, see, like this. If this structure, what these different जो मैंने यहाँ पर boxes बनाए, what this is called, I say that this end is the 5' and this is the 3'. जो हमने पहले कहा कि mRNA it is found in the direction of 5' to 3'. This end, it is called as cap. This end, it is called as the tail. This is called as the cap and this end is called as the tail. Now this cap जो है, it consists of, it contains cap, it consists of this cap, it consists of methyl guanosine, methyl guanosine, methyl guanosine, guanosine triphosphate, which is written as methyl guanine triphosphate, methyl guanine triphosphate, 3 phosphate groups. Now this tail, it is called as, इसको हम क्या बोलते हैं, poly A tail. It is called as poly A tail, and here we will found here there are 200 to 300. There are 200 to 300 adenylic adenylic acid. So it is called as poly A tail. Here there are 200 to 300 adenylic acid residues, rather not acid, but these are the adenylic residues are present. So this is cap, this is the tail. Now this blackish box, this what is this? It is called as initiation. This box, it is called, sorry, this box, it is called as non-coding region. This is called as non-coding region. And this box, this, it is called as termination. It is called as termination. This is called as termination codon. It is called as, oh sorry, it is not, oh sorry, this region, it is also the non-coding region. Sorry beta, it is also called as non-coding region. Sorry beta, it is also the non-coding region. This is non-coding region, this is also non-coding region. Now I will mark this line, this, I will mark this line, this. Now regarding this, what is this? This line, it is the initiation, INITIATION, initiation codon. That is, it starts the coding, which starts the, it is this, which starts the formation of, protein chain. We started the formation of protein chain. What are these codon? These are AUG. This, yeah, I will write it in this different color so that it will become somewhat clear to you. AUG. Now this part, इनिशिएशन कोडन, व्हाट इज दिस, टर्मिनेटिंग कोडन. यहां पर हम क्या देखते हैं, कोडन जो है दे एंड्स, टर्मिनेशन कोडन, and there are 3 termination codon. Yeah, we can simply say these are the termination codon that stops, जो stop कर लेता है, या that terminates, that stops codons and they are of 3 types. They are of 3 types. What are these 3 types? We have got, I will write them in this, UAA, UAG, and we have got UGA. These are the 3 codons that stops this termination, and these are of 3 types. Now what is this region? This region up to this is the, what we will call as this is the coding region. This is the coding region, and it consists of the codons. It consists of the codons which take part in the formation of the amino acids. So if we draw the structure, we will find this structure. This it starts from the 5' to 3'. This is the cap. This cap, it consists of the methyl guanosine triphosphate, MGGP. Then we have got the non-coding region. Then this small box, it is the initiation codon which starts the formation of protein chain, which codon AUG. In some books you may find one more, that is this called CUG. Now बीच वाला जो बड़ा, it is the coding region. This is the main part where we will see that it consists of the codes for the formation of mRNA. Then again we have got the termination codon. These are the codons that stops this coding, and they are of 3 types, UAA, UAG and UGA. Then we have got the non-coding region, and then we have got the tail, which is called as the poly A tail. Here what we will see, there are about 200 to 300 adenylate acid residues are present. This is a somewhat diagram. Now in case of eukaryotes, we are not taking part of the prokaryotes. Now, in case of eukaryotes, if we talk of in case of eukaryotes, what we will see in case of eukaryotes, the mRNA is not directly, the mRNA is not directly formed from DNA. From DNA. In case of eukaryotes, what we will see, mRNA is not directly formed from DNA. What we will see from DNA, what happens? From DNA, what happens? The reaction is like this. We will find hnRNA, and then from hnRNA, we will find mRNA. Now, what is this hnRNA? This hnRNA is heterogeneous nuclear RNA. hnRNA, it is the heterogeneous nuclear RNA, and this heterogeneous nuclear RNA, it is the precursor for the formation, precursor for the mRNA. So this hnRNA, it gives rise to the mRNA, and it is called as the precursor nRNA. Now, what is this hnRNA? What is this hnRNA? It is simply the heterogeneous nuclear RNA which represents what does it represent? It represents the split gene. It represents the split gene. Now, in this hnRNA, it consists of two types of sequences. Rather, we will find two types of sequences. Two types of sequences are found, and what is that called? एक को हम बोलते हैं, number one is called as exon, and second one is called as the intron. First one is called as the exon, and another one is called as the exon. And एक बात मैं यहाँ पर भूल गया, hnRNA का जो कॉन्सेप्ट है, it was given by Philip Shaw. Now, see the diagram. What will happen? Now, what happened? In this hnRNA, we will find two sequences of this, what we call as two sequences are found there. First one, this one. This one, it is called as exon. This is called as intron. This is called as exon. This is the intron. This is the exon. This is the intron. So we will find sequences of two types of these sequences. Now, what are these exons? These are basically they are the expressed, what we call as the sequences. They contain genetic information. They are what we call as basically they are expressed, they can be expressed rather, they are expressed sequences, and they contain genetic information, and they contain genetic information. This part, this इंट्रून की अगर हम बात करेंगे, इंट्रून, this part, they are not expressed, they are intervening sequences. इनको हम बोलते हैं intervening sequences, and they do not contain genetic information. They do not contain genetic information. What happens now when we will see if these are removed? These what we call the intron, we do not what we call as they do not contain genetic information, they are removed, and that removal of introns is called as, now the removal of introns is called gene splicing, or this is called as RNA splicing, LICING. Now since these are intervening sequences, they do not contain genetic information, they can be removed. So what happens, the structure कैसा बन जाएगा, like this. Again, this is the axon, this is axon, this too is axon. Sorry, I will write with this red pen so that it will become somewhat clear to you. This is axon. So here we will see the axon जो हैं, which are these expressed sequences, they are lying side by side. Axon, EXONE, axon, etc. So these are lying side by side. So a mature or sequenced mRNA